The current modern Western world has many privileges and advantages. However, this modern society also has its modern issues. One of these issues is the excessive daily intake of sugars, which is correlated with several lifestyle diseases such as obesity, diabetes, and cardiovascular disease. Two examples of countries where people eat way too much sugar are the Netherlands and the United States of America.
Hello.
The sugar intake of the Netherlands in comparison with America is shown. The daily recommendation of sugar intake is 90 grams a day, but in the Netherlands the average consumption is 102.5 grams of sugar a day, whereof 58 grams is glucose. This means that the average Dutch person eats 21 kilograms of glucose each year. In America, the consumption of sugar is also too high, 126.4 grams of sugar a day. 25% of the total sugar intake consists of sucrose. Examples of products which are consumed a lot and contain a lot of sugar are soda, cookies, chocolate, ice cream and candy. One of the main differences between glucose and sucrose is the difference in structure. Glucose is a monosaccharide and consists of 6 carbon atoms, 12 hydrogen atoms and 6 oxygen atoms. Sucrose, on the other hand, is a disaccharide and contains a glucose and a fructose molecule which are bonded together. This means sucrose has 12 carbon atoms, 22 hydrogen atoms and 11 oxygen atoms. Because of the fact that sucrose is a disaccharide, it first needs to be converted to monosaccharides before it can be used as energy by the body. However, when a person eats pure glucose, the monosaccharides are immediately present and after entering the body, glucose can immediately be used as an energy source in the body, because it does not have to be converted. These differences in structure led to questions such as, do these types of sugars really differ in the way they are used as energy in the body? To investigate this, the following research question is formulated. What is the effect of sucrose versus glucose intake on the respiratory quotient, energy expenditure and fat oxidation and carbohydrate oxidation as a percentage of energy expenditure between two persons resting for 1.5 hours? Along with this research question came the hypothesis: the processes of respiratory quotient and energy expenditure will reach peak levels sooner in subjects whose intake was glucose in comparison to subjects whose intake was sucrose. Energy expenditure after glucose intake will consist mostly of carbohydrates and little to no fat, and the energy expenditure after sucrose intake will consist less of carbohydrates and a bit more of fat. To answer this question, the next study design was created. The study design will be a double-blinded randomized controlled trial. The subjects are 2 females who are 18 and 24 years old and both study health sciences at the University of Maastricht. They will be tested in fasted state and standardized in the amount of sleep the night before. They will go to bed at 12 p.m. and wake up at 9:30 a.m. No caffeine or alcohol intake is allowed in 48 hours before the start of the experiment, and just before the start of the experiment, the subjects drink half a liter of water. Their physical activities before the experiment starts should be as limited as possible and registered in an activity logbook. Furthermore, the subjects are given the opportunity to urinate before the experiment starts. At 11 a.m., the students have to lay down in bed for 30 minutes for a baseline measurement. After 30 minutes, the students are double-blindly given a solution of 300 milliliters of water with either 75 grams of glucose or with 75 grams of sucrose. One person will arrange which participant drinks which solution. After the intake of water with dissolved carbohydrates, the subjects have to lay down on a bed for 1 hour straight. During this hour, they are not allowed to have any form of distraction or physical activity. The temperature in the room is kept at a constant 24.5 degrees Celsius and the subjects are linked to an indirect calorimetry machine which measures the amount of used oxygen and the expired CO2. This can be used to calculate the respiratory quotient, which is the ratio between CO2 produced and O2 consumed. Because the oxidation of protein is usually very small, protein oxidation was not considered in the calculation of RQ. The RQ represents the energy expenditure, fat oxidation and carbohydrate oxidation, and the percentage of energy expenditure derived from carbohydrates or fat in kilocalories. Up next are the results and the explanation of these results. In our hypothesis, we stated that glucose intake will lead to earlier peak levels than sucrose intake, since sucrose needs to be converted to glucose and fructose first, and fructose has a lower postprandial insulin response than glucose. This is in contradiction with our results. As you can see, the respiratory quotient is higher for the sucrose intake than for the glucose intake. This is contradictory to our hypothesis. However, there is an explanation for this. Fructose is known to be more potent than glucose as a stimulator for the novel lipogenic enzymes. The extra ATP requirements linked to the conversion from fructose to glucose in the liver and the energy cost of the novel fatty acid synthesis from glucose explains why the RQ is higher for the sucrose than for glucose. The results of the energy expenditure, which is the sum of carbohydrate oxidation and fat oxidation, are not in line with our hypothesis either. Sucrose intake results in an earlier peak in energy expenditure than glucose intake. We can explain this if we look at what happens in the body after glucose intake. After glucose intake, the insulin level in the blood rises, which results in a higher uptake of glucose in muscle cells, liver cells and adipose cells. The glycogen storage is restored in muscle and liver, and some fat is formed as well. So intake of glucose results in a higher storage rather than a higher oxidation. The body gradually shifts towards a state of more carbohydrate oxidation rather than a fat oxidation by increasing carbohydrate oxidation and decreasing fat oxidation. Because of fat oxidation results in more energy as compared to carbohydrate oxidation, the decline in energy expenditure can be explained. The fact that energy expenditure rises after sucrose intake can be explained by the fact that conversion of fructose requires some energy, as we also saw in the results on respiratory quotient. This figure shows that in the beginning, the first 10 minutes, after sucrose intake, fats are used as the source of energy. Carbohydrates are also used in this stage but increases with time. After 20 minutes, carbohydrates are the main source of energy expenditure after sucrose intake. However, this is not in line with findings in previous literature on this topic and our hypothesis. This figure shows that after glucose intake, the main source of energy expenditure in 60 minutes is fat. Carbohydrates are also used, especially after 20 minutes, but this source of energy is significantly less used than fats. As for the energy expenditure derived from fats and carbohydrates after glucose intake, these findings are also not in line with findings in previous literature on this topic and our hypothesis. The reason why the results are in contradiction with our hypothesis might be explained by the small scale of the research and the physical differences between the subjects, like age, height, weight, and muscle mass and muscle type. Subject A does high volume low intensity training, so the subject developed a lot of type 1 fibers. These fibers are the slow twitch fibers, also known as red muscle. These fibers have a greatly increased number of mitochondria to support the oxidative metabolism and a large amount of myoglobin that speeds the oxygen transport to the mitochondria. But subject B does low volume high intensity training. This means the type 2 fibers are more developed than the type 1 fibers. The type 2 fibers are fast twitch fibers, also known as white muscle, because of a deficit of red myoglobin. The fast twitch fibers have large amount of glycolytic enzymes for rapid release of energy by the glycolytic process. They have less mitochondria because the oxidative metabolism is secondary. To answer the research question, we can conclude that the respiratory quotient after sucrose intake increases more rapidly and to a higher extent than after glucose intake. The overall energy expenditure after glucose intake is lower in comparison to the expenditure after sucrose intake. After sucrose intake there is more carbohydrate oxidation than fat oxidation, while after glucose intake there is more fat oxidation than carbohydrate oxidation. As we already discussed, this is in contradiction with our hypothesis and previous literature, and therefore more research with a greater subject group with greater diversity is needed. Thank you very much for watching.